Welcome back to Further Reading with me, Dr. Susan Heavey from UCL. Today we're going to be looking into a really interesting paper on assembloids. If you've not heard of assembloids before, or maybe from the term, you can guess that there's something along the lines of organoids or tumoroids. Well, A, you'd be right, and B, sit back, relax, and let me take you through this new paper that looks at assembloids in a really interesting context. As usual, I'll start with a quick figure-by-figure overview of exactly what the authors did and what they showed, and then we'll get into some bigger picture discussion after that. So feel free to jump around using the chapter feature, and I'll see you in a second for figure 1. Figure 1 sets the foundation for the whole paper. The team took biopsies from 3 stomach regions, fundus, body, and antrum, and grew them into organoids. Despite being cultured in the same conditions, each organoid line kept its original regional identity. Panel A shows how organoids are derived from patient biopsies. In Panel B, native tissues display expected regional markers, IRX3 in the fundus or body, PDX1 and GAST in antrum. In panels C to E, all 3 regions form similar-looking spherical organoids. But immunostaining in panel F reveals distinct marker expression patterns. Antrum organoids express PDX1, GAST, and MUC6. Fundus body organoids express IRX3, GHRL, and PGC. There's no ATPase beta, so no mature parietal cells yet. And in panels G to H, RNA-seq PCA plots confirm the same story. Antrum organoids separate cleanly from the fundus or body, mirroring tissue regionality. The key message here is that even before building the complex multi-regional assembloids, the authors show that gastric organoids already sort of remember where they came from. This is a crucial step for assembling region-accurate stomach models. Figure 2 demonstrates how simple gastric organoids can be fused into more complex 3D structures called assembloids. Panel A is a schematic, a single region assembloid or SRA and multi-regional assembloid MRA. These are formed by embedding whole organoids in floating collagen 1. By day 4 in panel B, we can see that organoids begin to fuse inside the collagen gel. By day 10, in panels C and G, SRAs and MRAs form tubular structures with clear lumens. In panel D, whole mount imaging shows a continuous shared lumen with openings and budding protrusions. In panel C, SRAs secrete MUC5AC into the lumen, confirming gastric epithelial identity. In panel F, apicobasal polarity is established. F-actin apical, integrin, beta-4 basal. In panel H, TEM shows mature epithelial features, microvilli, desmosomes, and mucin granules. In panel I, MRAs preserve region-specific identity, PDX1 positive endocrine cells at the antrum end, PGC positive cells only at the fundic end, and high MUC5AC throughout the structure. The key message from this figure is that assembloid formation allows gastric organoids to organize into a structured, polarized, multi-regional tissue with early functional characteristics, something that single organoids simply cannot achieve. On to figure 3 now. Multi-regional assembloids, or MRAs, shift transcriptomes towards more mature gastric states and reveal region-specific crosstalk pathways not present in simple organoids or SRAs. Figure 3 digs into the molecular consequences of assembly. Using this RNA-seq across all conditions, so tissue, organoids, SRAs, and MRAs, the authors show that MRAs trigger major transcriptional changes that reflect tissue-like maturation and new signaling interactions. In panel A, PCA, including tissue, shows all in vitro models cluster separately from native stomach, but SRAs and MRAs sit closer to tissue than organoids. In panel B, PCA without tissue shows distinct clustering of organoids, SRAs, and MRAs, confirming each system has a unique transcriptional identity. In panel C, we see a key gastric marker shift. PDX1 remains antral specific. Endocrine markers GAST, SST, GHRL appear appropriately in MRAs. Parietal cell genes like ATP4A and B are low overall but detectable, aligning with early parietal emergence in later figures. Stem cell markers LGR5 drops in MRAs, reflecting differentiation. In panels D to E, ligand receptor analyses show new crosstalk pathways activated only in MRAs, including Notch DLL4 signaling in the antrum, EGF family ligands like AREG and HBEGF, BMP2 linked to parietal cell differentiation, and these pathways are more pronounced in MRAs than SRAs, meaning multi-regionality, not just 3D structure, drives maturation. The key message you should take away from this is that MRAs are not just bigger organoids. They activate unique interregional signaling networks that push gastric cells towards mature, region-appropriate identities. Now, Figure 4 is the big functional breakthrough of the paper. It proves that MRAs aren't just architecturally complex, they actually work like real stomach tissue, producing acid and responding to physiological and pharmacological cues. In panel A, mass cytometry via TOBIS-MC clearly separates MRAs from organoids based on signaling profiles, indicating a matured cell state shift. In panel B, stem cell markers SOX9, LRIG1, and STATHMIN1 drop in MRAs, while differentiated markers like GAST, SST, ghrelin, and MUC5AC are increased, confirming functional differentiation. A key highlight here is that ATPase beta, or a parietal cell marker, is strongly upregulated in MRAs, especially in the fundus region. In panel C, protein analysis shows the glycosylated functional form of ATPase beta is significantly higher in MRAs than in organoids or SRAs, an essential step for acid secretion. Then in panels D and E, immunostaining quantifies ATPase beta positive parietal cells. Erlotinib, which is an EGFR inhibitor, reduces these cells and lowers ATP4B mRNA, confirming dependence on EGF and BMP signaling. Then in panels G and H, they do a functional acid assay for that saccharidine orange. Histamine increases acidity, so higher red to green ratio in the MRAs, and omeprazole blocks this response. SRAs show almost no functional change, proving multi-regionality is required. In panels I and J, single-cell time-lapse imaging shows canalicular acid secretion patterns in fundic parietal cells, with maximum signal after histamine stimulation. This is a hallmark of functional parietal physiology. Overall, Figure 4 shows the first convincing evidence that these gastric assembloids generate functional parietal cells capable of regulated acid secretion, a feature never achieved in primary human gastric organoids before. Figure 5 demonstrates the power of the MRA system for patient-specific disease modelling. Using biopsies from 2 children with PMM2 HIP-KD IBD, the authors reconstructed the disease phenotype in vitro, including abnormal growth, mucin defects, and impaired acid-secreting machinery. Wow. In panel A, organoids derived from PMM2 variant tissue look similar to healthy controls but show more budding structures and are generally larger. In panel B, quantification confirms significantly increased organoid size, especially in antral and polyp-derived lines. In panels C and D, Ki67 staining shows comparable proliferation in isolated organoids, but focal hyperproliferation in PMM2 MRAs, especially in the antral region. In panel E, polyp-derived organoids show whole PDX1 positive buds packed with Ki67 proliferating cells, matching the patient's hyperplastic pathology. In panel F, histology of PMM2 MRAs reveals hyperplastic cell aggregates on one side, mirroring patient biopsy architecture. So cool. In panel G, TEM shows disease-typical mucin defects, thinner, less electron-dense mucin granules indicating impaired maturation. In panels H and I, PMM2 organoids and MRAs have reduced MUC5AC protein, and unlike healthy controls, they fail to upregulate MUC5AC upon assembly, consistent with decreased mucin secretion seen in patients. Then in panels J and K, the parietal cell ATPase beta pathway shows clear dysfunction. PMM2 MRAs have reduced ATP4B expression, and WES analysis shows decreased glycosylation of ATPase beta, producing an abnormal ratio of precursor versus functional protein. This aligns with the PMM2 role in N-glycosylation. Finally, in panels L and M, functional acid secretion assays show no histamine-induced acidification in PMM2 MRAs, confirming loss of functional parietal cell activity. Overall, Figure 5 shows that patient-derived MRAs don't just model anatomy, they recreate the core biochemical defect, that's N-glycosylation failure, and its downstream epithelial consequences, offering a true in vitro disease model for PMM2-related gastric pathology. Okay, so I think with a paper like this, it's worth taking a little bit of a zoom out and placing this methodology in context, because we've just seen an awful lot about assembloids, but what actually are they and how do they compare to perhaps more widely known and familiar terms like organoids and tumoroids? So let's start with a definition real quick. What are assembloids? Well, they're 3D multicellular constructs formed by fusing or integrating multiple organoids or tissue types, enabling interregional or interlineage interactions. They model cell migration, axon projection, synaptic integration, cross-tissue interactions, these kind of things that aren't accessible in single organoids. They're often used in neuroscience, but also cancer biology, immunology, gastroenterology, reproductive biology, and plenty more. So I've tried to track down when this term originated. It seems to have first been described as a multi-region fusion assembloid in 2017 by Pasca, Noblich, and Park. But the term assembloid itself was, I think, later coined by Pasca, and this was to describe fused functional neural spheroids that integrate and form circuits. So clearly, assembloids can do some pretty cool things, but why would you want to use these versus perhaps the more widely understood organoids or tumoroids? So some quick definitions here. Organoids are self-organizing 3D models of a single region or germ layer lineage. They have limited intertissue communication, whereas assembloids integrate multiple organoids or cell types to model emergent biology. So things like migration, vascularisation, immune tissue crosstalk, and so forth. Tumoroids are the kind of oncology look at this. So they're organoid-based cancer models that focus mostly on tumor microenvironment. Things like invasion, drug response can be incorporated into multi-lineage assembloids if you wanted. So you can get these sort of tumoroid assembloids, hybrids that in my mind sound pretty cool, and I'm quite tempted to get into these. If you want to read some really nice recent studies on this, I'll drop some links in the description below. There was a paper on multi-region neural assembloids, so things like cortex, striatal, corticospinal, retinothalamic, model circuit formation, interneuron migration, and long-range projection, super nice. You've also got multi-lineage assembloids, so integrating endothelial cells, microglia, or immune components to study neuroimmune and vascular interactions. And there are also, of course, non-neural assembloids, so like liver, kidney, gut, brain, bladder, expanding applications to organ-organ communication and disease modeling. Super interesting papers here. If you'd rather a kind of broader look, I'll link some reviews down below as well, which actually give you kind of a historical perspective, origins of the actual term, and also further comparison between different 3D model types. Enjoy. So it's without a doubt that even these super cool assembloids are missing physiological complexity. So even though they integrate multiple lineages or regions, they still lack the full cellular diversity present in real tissues. Nature's 2021 Outlook notes that these models capture interactions between specific cell types but remain far from whole organ complexity. They're missing vasculature, true stromal compartments, and full immune integration. Similarly, we see that dozens of assembloid types are being used, but most are simplified multi-organoid composites. They're not physiologically organs. Secondly, they still have quite a restricted lifespan and scale. So a couple of reviews, one in development and one in cell stem cell, highlighted that assembloids can only be maintained for a few months, and their regional fusion efficiency and long-range connectivity vary widely across labs. They often model early developmental events pretty well, but struggle with later maturation states. Now, a few months is still a pretty decent lifespan, certainly against the patient-derived explants that we use in my team, which can only really last for a scale of weeks or even days before they get a little bit wonky. But still, months is not useful enough. It would be great if we could get a model that could go further along years and we could see kind of long-range connectivity. They still have limited mechanical and systemic context, so nature's outlook makes it pretty clear that assembloids remain static systems. There's no circulatory flow, no hormonal signaling, and none of the mechanical forces that would be present in vivo. This limits their value for questioning things around metabolism, immune circulation, or organ-organ coordination. Other models still excel in different areas, so explants, which we actually use in my team, are fresh from human or animal tissue, and they fully preserve the native architecture. They have a natural ECM, vasculature, and stromal positioning, and they provide immediate clinical relevance for only short-term studies. So you've got some pros versus some cons here. Then from patient-derived explants, you can actually match the in situ disease environment. Usually it would be in oncology studies, and so you'd be looking at the tumor microenvironment, and that will directly match the patient that they've come from. And these can outperform organoids and assembloids for drug response fidelity in the short term because nothing's dissociated. PDOs, or patient-derived organoids, are good for medium throughput drug designs. They retain some patient-specific genetics, but they're single lineage, so they lack vasculature and stromal complexity, which tumoroids or assembloids attempt to address. Then PDX models, or patient-derived xenografts, these are models with patient-derived tissue that have been implanted into xenografts, so usually mice, and they're the gold standard for in vivo relevance. They provide vascularisation, organ-level context, partial immune interactions, but they're ethically challenging because of the animal work aspect. They're super expensive, they have limited throughput, and at the end of the day, from a biological point of view, a mouse is still not a human. From the 2025 review in Cells, organoids, assembloids, and tumoroids are situated as complementary, not competing, with each capturing different levels of biology. So certainly from where we're at right now, still, I would say that different models could be the right choice depending on what your biological question is. And we definitely don't have a one-size-fits-all approach yet. If assembloids want to be that in the future, then they still have a few things they need to improve on. So moving forwards, maybe in the next 5 to 10 years, wouldn't it be great if we could see vascularized and perfused assembloids? So reviews in Cell Organoid 2024 describe multilineage assembloids incorporating endothelial cells and blood vessel-like structures as a key frontier. Perfusion would enable nutrient delivery, waste removal, and more realistic gradients. Cell Stem Cells 2024 review highlights systems combining organoids with microglia, immune progenitors, or myeloid cells, enabling models of inflammation, neuroimmune crosstalk, or tumor immunology. Larger-scale multi-organ interacting assembloids would be amazing. There are some gut-brain or tumor stroma or liver immune assembloids already emerging, and these could become organ axis models approximating systemic physiology. But a key thing to consider is standardization and reproducibility. So current systems vary in terms of fusion efficiency, regional patterning consistency, long-range connectivity, and better patterning controls will allow cross-study comparisons and clinical translation. Finally, future work may use long-term culture systems, mechanical cues, or metabolic engineering to push assembloids beyond embryonic-like states towards adult phenotypes. Maturation and aging is a fascinating area of biology that assembloids could help us to look at in the future, but we're just not quite there yet. As we wrap up, it's worth stepping back and asking how all of this fits into the context of the paper that we've been discussing. The authors here take a thoughtful approach. They choose a method that's tractable, reproducible, and appropriate to the questions they're asking. And even though assembloids are incredibly powerful for modeling multilineage interactions, migration, and tissue-tissue crosstalk, they're not always the right tool for every study. So could these researchers have used other models? Possibly, but each alternative comes with trade-offs. Patient-derived explants, or PDEs, preserve native architectural and stromal complexity, but they're short-lived and low throughput. PDOs, meanwhile, are genetically accurate, but miss the interregional or immune components that might be needed. PDX models offer vascularisation and systemic context, but with huge costs, long timelines, and limited scalability. Seen against that backdrop, the author's choice of model makes sense. It plays to the strengths of organoid-based systems, controlled conditions, clean perturbations, and enough cellular diversity to answer the mechanistic questions at hand, all while maintaining the kind of reproducibility that assembloids can sometimes struggle with when fusion efficiency or lineage proportions vary between batches. And that's where the field is heading. Assembloids aren't here to replace these classical systems, they're here to bridge the gap between them. As they gain vascularisation, immune integration, and more mature phenotypes, they'll increasingly allow us to model the things that are currently split across PDOs, PDEs, and PDX models. We're still early in that transition, but the trajectory is clear. Assembloids will continue to evolve into a platform that captures more of the physiology reality that we need for clinical relevance, without giving up the control that makes reductionist systems powerful. If you want to dive deeper into how these molecular or developmental processes are interpreted in practice, I've linked another video at the end of this one. It's my walkthrough of the single-cell immune dysregulation paper, which pairs nicely with this discussion and shows how these frameworks actually get applied to real data sets. Thanks for watching, and I'll see you next time.